All right, welcome to our lecture on enhanced biomaterial design and production in a synthetic biology framework. My name is Maddie Paris, and I am joined today by Matt Lima and Alex Pellinger.
All right, to start with a brief agenda of our lecture, we will start with some background information on biomaterials as well as a look of key players in the development. Then we will look at current approaches, uses, and technology, and we'll discuss how biomaterials are widely used today. And then we'll move to the future and we'll look at more advanced uses of biomaterials and techniques that are still being developed. So to start with the background information, I'm going to be focusing on four main areas. First, the timeline and the important players, the chemical structure categorization, and the interaction categorization. So biomaterials have been around for ages. The first major development in biomaterials was in 1829 when H.S. Leverett studied metal implants in canines. Then in 1870, Joseph Lester introduced aseptical surgical techniques, revolutionizing surgery. In 1886, each handsman used metal plates for internal fixation within the human body. And in 1931, Smith Peterson developed a metal cup for hip implants. In 1947, the first paper was written discussing polyethylene as a synthetic implant material. And in 1967, Dr. Jonathan Cohen offered the first definition of biomaterials. From 1970 to present day, we've been experiencing a massive biomaterial and synthetic biology boom, and we're only learning more. The categorization of biomaterials can be split up into two main categories. First, we have the chemical structure category, and then we have the degree of interaction with biological environment categories. So the first type of chemical structure is the metallic structure. The first metal alloy developed for human use was the vanadium steel, which is typically used to make bone fracture plates and screws. And biocompatibility is very important in metallic implants because the metal can potentially corrode. Then we have ceramic biomaterials, which are extremely hard. In fact, the hardness of other materials are typically tested against ceramics, and these can be inert, semi-inert, or non-inert, which gives them a wide array of uses. Then we have polymeric materials, which are widely used in disposable medical supplies, prosthetic materials, dental materials, and more. And one of their main advantages is the ease of manufacturing. They can produce various shapes, which makes them very useful. Then we have composite materials. Some applications include dental fillings, dental filling composites, bone cement, orthopedic implants with porous surfaces. Then we have biodegradable materials, which can be broken down by natural processes, and they are gradually absorbed by the body and do not cause permanent foreign body reactions. Then our second set of categories are the degree of interaction with the biological environment. We have biotolerant materials. These materials are separated from surrounding bone or tissue by a layer that is typically induced by the implant releasing monomers, ions, corrosion products. Almost all synthetic polymers and most metals are in this category. So these materials are tolerated by the body, but they're not necessarily inert. Then we have the bioinert category, and these implants or biomaterials are in direct contact with bone and tissue. However, there's no chemical reaction between the two, and they're considered inert because there's no chemical reaction. Examples of these biomaterials are alumina, zirconia, titanium, things like that, as well as carbon. We have bioactive materials, and there is a distinct interaction between the implant and the surrounding tissue. The part of the tissue typically binds to the implant, and these materials that are in this bioactive class are calcium phosphate, vitroceramic, and whatnot. Then we have bioresorbable materials, which are materials that the body ends up degrading or absorbing, and they're of interest in clinical applications where you can't operate on a patient to remove the implant. And some biomaterials in this class are tricalcium phosphate and polyl lactic acid. And then at the bottom, we just have a few photos of different biomaterials. We have a tooth, which has a metal base and typically a ceramic top. We have a hip implant, which can be ceramic, metallic, polymeric, composite, could be anything. And then we have like a heart monitor that is implanted within the body.
Hello, my name is Alex, and today I'll be talking about current biomaterials used in an industry and techniques used to produce them. I will discuss tissue engineering, drug delivery systems, immune-safe materials, electrospinning, powder metallurgy, electrodeposition, and electric discharge machining, to name a few. First, we'll look at tissue engineering. As you can see on the slide, tissue engineering typically starts with stem cells, which are then separated and proliferated inside a petri dish. These are then combined with a hydrogel, which we'll talk about in a second, and then the solution is injected into either a bone or cartilage. Tissue engineering is typically used to repair damaged cartilage and bone tissue, as just mentioned. The tissue is created from biocompatible scaffolds that can successfully transport cells and maintain the growth of the tissue. Hydrogels, as seen in the figure, are used as they are biocompatible, non-toxic, porous in nature, and have a unique similarity to the cellular matrix of the body. Next, we'll look at drug delivery systems. They are typically made up of polymers, lipids, proteins, and peptides. Also, currently, DNA building blocks are becoming a viable option for creating 3D nanostructured drug delivery systems. These biomaterials listed are created to have a controlled release of drugs as a response to changes in the body. Typically, these changes will be a change in temperature, pH, or just general physiological symptoms. Third and lastly, we can look at immunosafe materials. Newly developed immunosafe materials such as modified polymers and lipids, photoactive materials, pH-sensitive materials, and other reactive groups have helped further creation of vaccines and development of immunotherapy. However, both the polymers and hydrogels listed before with drug delivery systems and tissue engineering could also fall under immunosafe materials. These materials can aid in the re-engineering of immune pathways, which is extremely important in healthcare. Now I will go into a little bit about production techniques. First, we'll look at electrospinning. Electrospinning is typically used with synthetic biomaterials like lipids and polymers and involves an electrostatic charge along with a spinner to essentially 3D print scaffolds and fibers. So as you can see, you have the polymer in a syringe where a high voltage is passed through it and the spinneret spins it around to essentially create the structure that you want. On the other hand, powder metallurgy is based on metals as a biomaterial, typically titanium alloys, and can create porous materials and scaffolds similar to electrospinning. It can also create parts that you want to as well. In this process, metal powder is added to an additive and then compacted down into a mold where it is fired up and sintered. Then it is shipped off as a final product. However, typically there will be a finishing process before this, which we'll discuss right now. Looking at the first surface modification technique, electrodeposition involves depositing a protective nanolayer on top of a material. Similar to coating metals, the process involves electrodes and electrolytes to coat the biomaterial. This process can improve wear resistance, corrosion resistance, and increase biocompatibility. Next, we'll look at electric discharge machining. This machining produces a large number of sparks in a very short period of time in order to remove material from the biomaterial surface. This creates a biocompatible layer and enhances the hardness and corrosion resistance of the material, as seen in the diagram above. However, bubbles and eroded debris can be created, which may mess up the discharge machining. Finally, we'll look at thermal spray coating and surface finishing. Both of these are used to alter the surface of a biomaterial and increase the wear resistance and mechanical properties of that material. Thermal spray coating involves a cathode and electric and gas flow, as well as to create a plasma torch which will melt a powder that will then coat the material. Surface finishing, on the other hand, can be many different things, but in this instance, we'll look at a special CNC milling head that turns and hammers chunks out of the surface of the material. As mentioned before, both of these can customize the surface of the material so that it's more biocompatible with the body and allows greater wear resistance and corrosion resistance, as mentioned before.
Now we'll talk a little bit about the future directions of biomaterial production and design, specifically focusing on 4 categories: biomimetics, engineered living materials, self-reproduction, and computational methods. Biomimetic materials take their inspiration from nature. The most basic building blocks are used to recapitulate natural materials through synthetic production methods, or to use these building blocks to incorporate enhanced function to produced materials. This is done by taking natural proteins from things like spider silk and dragonfly wings, characterizing these proteins and learning about their structure and function to then turn these into modules. These modules fall into 3 categories: structural, functional, and responsive building blocks. Structural blocks are often, but not always, based on repetitive protein sequences like silk and collagen. These blocks provide physical and chemical characteristics to the protein. Functional blocks can be components like cell adhesion peptides or proteolytic recognition domains. These blocks provide a certain function to many proteins, and responsive blocks are often derived from biological switches and impart a sensing capacity to proteins. Examples of responsive blocks are light-induced crosslinkers or small molecule-driven dimerization motifs. All these blocks can be mixed and matched in order to relay certain desired attributes to the completed and assembled protein. Engineered living materials pair living organisms with inert materials to give the final product the dynamic sensing and actuating abilities of living systems, while benefiting from the stability and consistency inert materials provide. An example of this is a living therapeutic that incorporates antibacterial producing organisms into a polymer hydrogel. The engineered spores produce the antibiotic upon sensing the pathogen. The result is an antibiotic patch that can sense the harmful bacteria and kill it when it's present. Another example is a living electronic device, which utilizes bacteria engineered to sense specific substance in its environment and output some readable signal as a result. These bacteria are put into essentially what amounts to a pill that can be both let in environmental cues and transmit a signal outside the body. The outcome is a system that can transmit data about the environment it is in and provide the ability to monitor health. Self-reproduction relies on the reduction of metabolic pathways to their most basic constituents, then purifying the biological components responsible for the production, and creating controlled reactions that attempt to circumvent the messy nature of biology. Purified enzymes, cofactors, cell extracts, and energy molecules are a few components that are incorporated into cell-free reactions. Some benefits of cell-free reactions are their ability to more quickly identify bottlenecks to the precise control they provide, and the avoidance of cellular toxicity that can arise when producing cytotoxic or large amounts of products. The goal of computational methods for biomaterial design is to be able to take a desired functional capability or material property and accurately predict how to create such a material from design to assembly and processing. One possible outlook is to correlate the materiome to other omics data sets in the hope that elucidating the relationship will result in a certain material property. Classification and optimization of biomaterials is often slow and resource intensive. If the materiome and transcriptome or proteome were linked, the design process could be much more deliberate, tunable, and efficient. The hope would be to start replacing some aspects of experimentation with modeling to improve the resulting material properties and reduce the intermediate steps to get there.
And then just to run through a quick summary, we first went through the background material and learned that biomaterials have been around for ages, but we are currently experiencing a technology and a knowledge boom. Then we went through current techniques and the current production processes and learned that they allow for the creation of biomaterials that can adapt and thrive in their respective environments. And then we went through novel techniques and learned about next steps and the fact that they will be a drive towards modularity and predictability for improving biomaterial behavior. Thank you for listening to our presentation. If you have any questions, please reach out. Our information is on the slide. Thank you.